Good morning, viewer. Biendas here. Here is the most amazing news for our viewers. Yeah, a crucial gene has been identified that clarifies the repeated emergence of gliding capabilities throughout the evolution of marsupials. Beautiful. People, people say when pigs fly to describe the impossible, but even if most mammals are landlubbers, the ability to glide or fly has evolved again and again during mammalian evolution. In species ranging from bats to flying squirrels. Yeah, how did that come about? In a study recently published in the journal Nature, a team of researchers led by Princeton University and Baylor College of Medicine explains the genomic and developmental basis of the patagium. The thin skin membrane that allows some mammalian species to soar through the air. We don't quite understand how novel traits and adaptation originate from a molecular and genetic perspective. We wanted to investigate how an evolutionary nobility arises, said co-corresponding author Dr. Ricardo Mallerino, assistant professor of molecular biology at Princeton. To better understand patagium evolution, the team focused on marsupials. That is because the ability to glide has developed repeatedly using similar anatomical changes in closely related marsupials like the sugar glider, a tiny marsupial small enough to fit in your pocket and popular as an exotic pet. The Ballot team led the genome sequencing for 15 marsupial species, determining the DNA sequences in both gliding species and their non-gliding relatives. Comparing those sequences revealed accelerated evolution near a gene called EMX2. What's interesting is that the sequence of the gene itself does not seem to be where the most relevant changes are taking place. Instead, the key changes are in short DNA sequences called enhancer that lie nearby the genome. It's those changing enhancers that after how and where in the body EMX2 is active, and that drives the evolution of gliding, said co-corresponding author Dr. Erich Lieberman Iden, professor of molecular and human genetics and director of the Center for Genome Architecture at Baylor. Understanding the underlying changes that happen at the genomic level to give rise to these convergent traits is important because it can tell us whether evolution is targeting the path of least resistance. You can have the same outcome but different paths to get there, said co-author Jorge Morenin, a graduate student in Murillo's lab. Next, the researchers wanted to test these ideas. To do so, they used one of the most unique characteristics of marsupials, their pouch. Marsupial joyers are born at a much earlier stage in development than typical mammals, said co-first author Dr. Olga Dukchenko, assistant professor of molecular and human genetics at Baylor and a researcher at the Center for the Theoretical Biological Physics at Rice University. Instead of continuing development in their mother's womb, they crawl into her pouch and stay there until they are ready to take on the world independently. The fact that they are right there in the pouch makes it much easier to study how individual genes like EMX2 affect the marsupial's development. BN's exclusive report.